So welcome to our virtual campus. Our first speaker will be Athma Pai. Athma is an assistant professor in the RNA Therapeutics Institute. She joined us in 2018, and before that, she was a Jane Coffin-Childs postdoctoral fellow in Chris Burge's lab at MIT. Today, her recent work on the kinetics of RNA processing will be the topic of her seminar. And after Athma, we'll hear from John Watts, who I'll introduce after we take questions following Athma's seminar. If you want to ask questions, please use the Q&A box, and we'll hopefully get to everyone. Athma?
Great. So thank you to Phil and Angela at the RTI for organizing this, and to the wider RNA community for both organizing this from other centers and also participating so eagerly. I see that we have, I think at current count, hundreds of participants on the Zoom call. So today I'm going to tell you a little bit about my lab's work in trying to understand the kinetics of mRNA processing. And I like to think about it with respect to how quickly we go through gene regulatory steps. Oops, there we go. Okay, so we're all familiar, of course, with the central dogma of molecular biology, where DNA is transcribed to create RNA molecules, which are then translated at the ribosome to create proteins. But we know that this, of course, is an oversimplification of the actual processes, where RNA doesn't magically get transcribed from DNA in just one step. But in reality, we know that each of these steps are made up of many specific molecular and biochemical mechanisms, such that the act of creating a mature mRNA molecule involves not just the process of transcription, but also steps such as mRNA splicing, 3' end processing, exporting that RNA, localizing it to the proper place, and then a whole host of mechanisms involved in translation. Now, each of these processes have been studied in depth for many years, and from that research, we know that they act together to cumulatively establish a steady-state transcriptome or proteome, but we actually know quite a bit less about how they're coordinately regulated. To be able to really understand how gene expression is regulated, I think we have to directly study the synergy between these individual processes, and that synergy likely happens through a complex dance of these molecular events, and in particular, a complex interplay of the relative rates at which these mechanisms are occurring. And that premise fascinates me, particularly with respect to the diversity of RNA molecules that comprise our transcriptome. And so while tracking the timescales of each of these individual molecular processes, you can imagine that there's a certain amount of time that's spent in each one, and that's regulated by some limiting factor. The changes in the rate or the time scale at any given step might result in different expression levels or different mRNA isoforms that are expressed. So this variation in individual rates, sorry for the animation difficulties here. So this variation in the individual rates of molecular events is what we ultimately see as steady-state variation in mRNA expression levels. But despite the centrality of the kinetics of each of these steps to create this transcriptome, we really have a very poor understanding of, one, how to track these rates at a genome-wide level, and second, how they coordinate to give rise to a desired isoform or transcriptome composition, and ultimately the desired phenotype in a given cell. So the steps of RNA processing are canonically thought to be mostly co-transcriptional, such that splicing, for instance, is thought to occur while the rest of the mRNA molecule is still being transcribed. They're also thought to be linear in their progression. Of course, you first transcribe the RNA, but then the sequence of events is usually depicted such that capping, splicing of all the introns, and then cleavage and release from the chromatin happen in exactly that order. But there's increasing evidence that the timing and hence the order of these events might specifically be regulated and could be regulated differently in different cellular contexts. So, for example, Daniel Larson's lab showed that for the beta globin gene, the splicing of a given intron can occur stochastically either before or after cleavage and release of the molecule from pol 2. When they looked at cells with a mutation in the U2AF core splicing factor, they found that the timing was shifted such that splicing occurred entirely post-release of the mRNA molecule. Now, interestingly, that mutation in the U2AF splicing factor consistently recurs in cancer cells, suggesting that that mutation may play a broader role in kinetic competition during cancer progression. Another one of my favorite examples is from Doug Black's lab, where they profiled RNA after cellular fractionation in macrophages and saw that the sequential waves of gene expression that occur after lipid A stimulation in the immune response may be gated by the kinetics of splicing and cleavage rather than transcription initiation cascades. So these examples and many others in the literature highlight the need to move beyond just characterization of steady-state transcriptomes, but more towards a dynamic understanding of mRNA biogenesis and maturation. So with that in mind, my lab aims to understand the molecular basis for transcriptome diversity, specifically by developing and applying genome-wide methods to measure the kinetics of gene regulatory mechanisms. And one of the first questions that we'd like to tackle along this process is, what are the rate-limiting steps for RNA maturation across different genes? You might imagine that the rate-limiting steps for isoforms of gene A might be transcription, where all the other processing steps occur relatively rapidly after the relevant sites become available. But instead, for other genes, transcription might proceed quite rapidly. Instead, the creation of a mature and productive mRNA molecule might instead be gated by the rate of splicing or the rate of cleavage on a given mRNA. And to tackle this question, the first challenge that we have is to actually develop high-resolution approaches that allow us to systematically and simultaneously measure genome-wide rates of key steps in the process of RNA biogenesis and maturation. And we're initially focusing on these steps, that of the rates of transcription elongation, the rates of mRNA splicing, and the rates of 3' end cleavage and polydenylation. So first, I'd like to tell you about a method that we developed for measuring the rates of mRNA splicing, and this was work that I did during my postdoc with Chris Burge. So first, how do we actually go in and measure the rates at which mRNA processing generally is happening? To do so, we need to be able to capture RNA molecules across a range of time periods, both before and after splicing, for instance. And the way in which we do that is we use metabolic labeling, specifically with a 4-thiouridine molecule, to incorporate 4-thiouridine into newly created RNA and be able to specifically capture this nascent RNA across a range of labeling time points, with the assumption that an earlier time point, such as a 5-minute time point, might give us a pool of molecules that still contains many introns before they've actually been spliced out, while longer time points would have more opportunity for splicing to occur and more molecules where splicing has already occurred. So looking at the rate of intron loss across these time points of labeling would allow us to measure the rate of splicing and specifically the half-life of intron excision per intron. And so the data that I'm going to talk about for my entire talk today is from 5 minutes, 10 minutes, and 20-minute incubations with 4SU. And this was in Drosophila melanogaster S2 cells with 3 replicates per time point. And the data was generated in Karen Edelman's lab when she was actually still at NIHS. So the way in which I'm drawing this really nice exponential decay to measure the rate of splicing on the right-hand side here, turns out is actually an oversimplification of the data we have, given the way in which our metabolic labeling protocol was done. In fact, because we actually did a continuous labeling over the entire time point, whether or not it's 5 minutes or 10 minutes or 20 minutes, the data that we have actually accounts for a range of lifetimes across the intron. So the graph that I showed you previously assumes that the intron was created exactly when the label was added and had the full 5 minutes to be spliced out, and so we can measure the rate at which over that 5 minutes the intron was spliced out. But instead, that intron could have been created at any point within our labeling period. In fact, it could have been created only seconds before we extracted the RNA at the end of our labeling time point. And thus, while this initial intron has a full 5 minutes to be spliced out, this final intron here actually only has seconds to be spliced out. And so comparing the rates from those 2 molecules would not quite be fair. To illustrate this, we can imagine that each of these given molecules here contributes about 8 RNA sequencing reads to our final data set. Now, the proportion of either exon-exon junction reads, which indicate spliced molecules, or intron-exon junction reads, which indicate unspliced molecules, that each of these molecules provide is proportional to both the rate of splicing and also the time that this molecule had to actually splice out the intron. So while it's easy to derive a rate of mRNA splicing, if we know which of these time points or these labeling lifetimes each of these reads came from, our actual data looks something like this, which is an amalgamation across distribution of lifetimes. So to account for this complexity in intron lifetimes and their relative contributions to our read pool, we decided to specifically model the data in terms of the probability of observing either unspliced, which are these pink intron-exon junction reads, or spliced reads, which are these blue exon-exon junction reads, given the possible distribution of transcript lifetimes that we actually sample for across our labeling time periods. To do so, we used empirical counts of either exon-exon or intron-exon junction reads, and derived a molecule that derived a computational model that derives the that relates the ratio of these junction reads to the probability distribution of actually observing the reads at a given splicing rate. So I'm not going to go into the math behind this model today, but I will point out that this model and the data is published, and we use this model to estimate the rates of mRNA splicing for a little bit over 25,000 introns in the Drosophila genome, with a median intron excision half-life of 1.96 minutes. And we find that there are a variety of factors, both genomic and trans-regulatory factors, that actually influence variability in splicing half-lives. So I encourage you to go and read this paper to get more details. Instead, for the rest of my talk, I'm going to focus on some newer work from my lab to specifically measure the rates of 3' end cleavage and polyadenylation. And this is work by a graduate student in my lab, Leslie Torres-Olua. And what we did was we realized that we can use nascent RNA sequencing reads and adapt our previous splicing rate model to estimate the genome-wide rates of 3' end cleavage from an actively elongating polymerase molecule. So the estimation of these rates is crucial for actually understanding the overall rate of mRNA maturation, because that's the final step in creating a mature mRNA molecule before that polyadenylated mRNA molecule can be exported to the cytoplasm and continue on in its functions in the cell. Furthermore, we know that the formation of 3' UTRs and the choice of different 3' ends can have many influences on the function and the lifetime of that mRNA molecule, where the choice of a different 3' UTR or the choice of a different polyadenylation site can influence mRNA stability, mRNA translation, nuclear export, mRNA localization, protein localization, and many other factors. Finally, we know that there's extensive alternative polyadenylation site usage in both the Drosophila genome and many other mammalian genomes that's likely regulated across different cellular contexts, different cell types, and mutated in the context of many human diseases. So there have been a few different models that have suggested kinetic regulation that actually influences the choice of these poly-A sites. One is that the rate of elongation or the speed of polymerase across the molecule might actually influence the usage of different poly-A sites. Where a polymerase is moving quite fast across this molecule, it might see both of these poly-A sites at the same rate and result in a longer 3' UTR. But if polymerase is moving quite slowly, you might actually use this upstream poly-A site before you even have a chance to transcribe this downstream poly-A site and result in a shorter 3' UTR. So this is a very intriguing model, but there's another component to it, and that involves a balance between both the polymerase speed and also the rate at which that site is actually cleaved at. So, independent of the speed at which polymerase is moving across that site, if you don't actually load factors onto this polyadenylation site and cleave it quite quickly, you might still have an equal opportunity to cleave at both of these sites. So the faster cleavage at this upstream site might result in shorter 3' UTR molecules, but the slower cleavage at this upstream site might actually result in more diversity of 3' UTR lengths and 3' UTR variation. Okay, so in order to be able to derive this computational model to be able to estimate the rates of 3' end cleavage, we first had to be able to actually differentiate between RNA sequencing reads that came from cleaved or uncleaved transcripts. And what we realized is that we had 4 types of molecules in our data. We had reads derived from mRNA molecules, pre-mRNA molecules that had not yet transcribed past the cleavage site. So they're denoted as before cleavage site molecules. We had reads from 2 different types of mRNA molecules that had transcribed past the cleavage site, both those that had transcribed past the cleavage site and not yet been cleaved, so or one contiguous molecule across that cleavage region, or those that had transcribed past the cleavage site and been cleaved such that there would be a mature mRNA, potentially even polyadenylated molecule in darker purple here, and reads deriving from the rest of the molecule that occurs from the pol 2 continuing to transcribe past the cleavage site. So we realized very quickly that we could not just differentiate between these purple and black molecules by only looking at reads that occur downstream of the cleavage site, since pol 2 actually in many cases transcribes for quite a bit of a distance past the cleavage site and does not actually end transcription until XRN2 loads onto the 5' end of this read-through molecule, and it's been proposed torpedoes the pol 2 off of the DNA. So now we're left with this idea of how do we still differentiate between reads that come from cleaved molecules and molecules that have the opportunity to be cleaved but have not yet been cleaved. So in order to actually start to understand RNA sequencing read patterns that might allow us to differentiate between these two, we did an in silico simulation of RNA sequencing reads from different 4SU labeling time points of nascent RNA reads. So each of these lines here is a different molecule simulated from a 5-minute labeling time point, 10-minute labeling time point, and 20-minute labeling time point. And these are all, again, simulated conditions with a 3 kb gene, a 5 kb read-through region after the cleavage site, a particular expression level, and a particular number of molecules, transcripts that are being transcribed. So on this plot here, left of this dotted line is the gene body, and right of the dotted line is the read-through region. So we can take each of these molecules and we can simulate a particular rate of cleavage. And here I'm simulating a 5-minute cleavage half-life. And with that rate of cleavage, we can see that in gray here are all the molecules that have not yet transcribed past the cleavage site. And then in purple and black here are the molecules that have transcribed past the cleavage site but have been cleaved at a particular rate. Then we can take these full pre-mRNA transcripts and actually put them through in silico the regeneration process or the library preparation process. The first step is to fragment these reads into short fragments, as is done in RNA sequencing library preparations. And then the second step is actually to perform a size selection. Now I'm zooming in on just a 1 kb region right around the cleavage site. So we can perform this size selection with a particular distribution of insert sizes that are used. And then we can select reads that fall within a 50 nucleotide standard deviation or 200 nucleotide mean insert size. And so what you can see here is that now you're starting to see a little bit of a difference between these purple molecules that have already been cleaved and these black molecules that have not yet been cleaved. And what we're specifically seeing is that there starts to be a depletion of fragments right in this region, right around the cleavage site, even across time points. We can then parse these down to 50 nucleotide paired end reads from each of these fragments, and you still see this depletion in reads right around this region. We realize that that depletion of reads is actually quite important because it represents an edge effect in RNA sequencing data where you're less likely to, you're less likely to sequence reads from the ends of molecules because you're more likely to lose those fragments during the actual size selection protocol in RNA sequencing library preparation steps. And what this results in, if you look at a metagene plot of all of the reads across these time points and across these transcripts that were simulated, is a pronounced dip in the amount of reads right at the cleavage site. And this dip is very specifically delineated where the reads that remain right across the cleavage site represent uncleaved RNA molecules, and the reads that are present in or are are depleted in this dip actually represent the cleaved RNA proportion. So this pattern right at cleavage site allows us to differentiate between uncleaved RNA molecules that have transcribed past the cleavage site and the total number of molecules, specifically the number of cleaved molecules that have transcribed past that cleavage site, but are informative to our understanding of how fast they've been cleaved. Furthermore, across time points, if we think about it, there's actually a difference in the extent to which this depletion of reads happens right at the cleavage site. So at earlier time points, you might have a lower dip relative to the total number of molecules on either site. But at later time points where more cleavage has had the opportunity to occur, that dip becomes more pronounced. So this is what this looks like in actual RNA, in nascent RNA sequencing data from the same Drosophila time points I was showing you previously. And we see exactly that. We see a pronounced dip in the coverage of RNA sequencing reads right at the annotated transcription end sites. And we see that that dip is more pronounced for later time points than it is for earlier time points. So this is not just only seen in the aggregate metagene plot, but you can also see this for individual cleavage sites at specific genes. It's noisier, but still quite predominant. So we used this information to derive a mathematical model to estimate the rates of pre-mRNA 3' end cleavage. And again, I'm not going to go into the math, but I'm going to point out three crucial assumptions that we had to make this model. First, we assumed, I think fairly, that the polymerase had to actually transcribe past the site of cleavage in order for a molecule to be cleaved. Second, we used reads that are present across this cleavage site as indicating uncleaved molecules, and we used reads upstream of the cleavage site as the total number of reads, the total number of fragments that could have been present that could have transcribed across the cleavage site. And finally, we can get informative reads from polymerases that have transcribed any distance from the cleavage site all the way to the end of the read-through region. And that's a region that we determine empirically from our nascent RNA sequencing data. So the last thing I want to tell you about our model development is that we went back to our simulations to understand whether or not the mathematical model that we derived actually worked accurately to estimate cleavage, to estimate the rates of cleavage. So we modeled a number of different contexts, including different insert sizes, different lengths of the read-through region, different labeling time points, different expression levels, and obviously different cleavage half-lives. And we're able to very accurately re-estimate the rates of mRNA cleavage across a range of different 3' end cleavage rates that we simulated. So using this model and the nascent RNA sequencing data from Drosophila melanogaster S2 cells, we estimated half-lives for a little bit less than 8,000 annotated cleavage sites in a little bit more than 6,000 melanogaster genes. We find that there's a median half-life of about 40 seconds, which is much faster than the median rate of mRNA splicing that we observed in our previous study. We do still, however, find that there is a large range of variation around this median cleavage time, where we have very, very fast cleavage rates all the way up to about 10-minute cleavage rates at a given particular site. And so the last question that we wanted to ask is, what actually underlies variability in the rates of mRNA cleavage? And so the process of actually performing a cleavage event is quite complex, involving many different both cis-acting and trans-acting genetic elements and regulatory factors. But the first thing we wanted to ask was, does the position or the sequence of the actual polyadenylation site influence the rate of cleavage? And we know that there are many sequences around the polyadenylation site that are important for binding various transacting factors to regulate cleavage occurring. And the most important one is the actual polyadenylation site, but there are many flanking ones that occur at various distances around the actual place at which cleavage occurs. So the polyadenylation site is thought to occur thought to be present about 10 to 30 nucleotides upstream of the site at which cleavage itself actually occurs. And there are about 10 to 11 known polyadenylation sites that are used predominantly in the Drosophila genome, 2 of which are used about 90% of the time. And this is a distribution of those sites relative to the annotated cleavage sites that we looked at. We see that the position of that sequence element relative to the site of cleavage actually does not influence the rate of cleavage at all. However, we see that the presence of particular sequences are enriched within the 10% of fastest cleaved sites. So we know that the actual the sequence context matters quite a bit. Furthermore, we also see that the more polyadenylation k-mers that you have present before upstream of a given site actually leads to faster cleavage rates. Thus, we know that having more of these sites might have an influence on the extent of binding of factors that regulate alternative 3' end cleavage, or could influence the rate of that cleavage in different ways. The final thing I want to mention is that we also were able to measure the rate of cleavage at alternative polyadenylation sites in the same gene. So we classified genes as either having single transcription end sites, alternative last exon sites where the polyadenylation site is in 2 different exons, or tandem 3' UTRs where the polyadenylation site is in the same exon and uses a different polyadenylation site, just extends the length of those exons. And what we observe is that when a gene only has a single transcription end site, cleavage occurs the fastest relative to genes that can have alternative transcription end sites that can be used in Drosophila S2 cells. Furthermore, we see that the proximal most site, the upstream most polyadenylation site, both for alternative last exons and for tandem 3' UTRs, tends to be cleaved slower than the downstream most or the distal most polyadenylation site, suggesting that there's actually some regulation of either distance or genetic architecture that influences the rate of polyadenylation, cleavage and polyadenylation that happens when an alternative decision is actually being made. With that, I'd like to summarize by saying that we were able to develop a robust computational model to estimate the rates of pre-mRNA 3' end cleavage. We find that 3' end cleavage rates are fast, but still quite variable. And we observed that gene distal alternative polyadenylation sites are cleaved faster than proximal alternative polyadenylation sites. We still have quite a bit of work to do to finalize this project, but we are working on characterizing and validating some of the factors that might contribute to variability in cleavage rates. Tying it back to the context of splicing rates that we have in the same cell type, and understanding the relative usage of splice sites and polyadenylation sites in a kinetic framework. And finally, identifying precise sites of cleavage using our nascent RNA sequencing data, and potentially even the rates of XRN2 cleavage of the read-through transcripts. So I'd like to thank everybody in my lab, and very specifically Leslie, who's actively still working on this project, funding sources, and mention that our lab is looking to grow and very actively recruiting postdocs. So please email me if you're interested at all in chatting about that. Thank you very much.
Athma, thank you very much. That was terrific. You have 2 questions so far. First, Matt Youngman asks, is successful upstream splicing required for the 3' cleavage to occur?
So there's some data that we haven't actually looked at quite a bit, but there's some data from long read sequencing data that suggests that if you don't properly splice upstream, you might not actually cleave at that site. What we're hoping to do is actually add a temporal component to that. We haven't yet looked at the rates of, for example, splicing of this upstream intron relative to the rates of cleavage at the downstream polyadenylation site. But I'm hoping that that analysis might provide some insights into your question.
Chapman Cavender asks, does the preference for distal poly-A sequence site over proximal PAS depend on the distance between the two sites?
We don't see an effect of distance between the two sites in terms of the relative rates at which they're cleaved at. The usage of both proximal and distal sites has been observed to be quite variable in different cellular contexts, and the cellular context actually, such as neuronal differentiation or immune stimulation, could influence a very predominant preference for either distal or proximal sites. And as far as I know, that preference is not dependent on distance, but I could be wrong there in specific cellular contexts.
We have three questions from people who are shy. So a first anonymous attendee asks, can you talk a bit about how the read-through region is determined empirically and the impact that any uncertainty in the estimate of this region would have on the computational model?
Certainly. So I didn't specifically mention this, but the way in which we actually calculate the length of the read-through region is using a program called Dogfinder, which is developed in Rayut Chalgi's lab. We run this on POL2 ChIP-seq data. And the reason we run this on POL2 ChIP-seq data is because we only want to look at how far the polymerase travels, but we actually don't want transcription elongation rates or XRN2 cleavage to influence our estimation of this read-through region. So we think that just looking at the position of POL2 is a better estimate of how far that polymerase is actually moving across it. So to your second question of how much the variability in our estimate of the length of the read-through region can actually influence our model, that's something we specifically looked at in our simulation data. We're still looking a lot more in depth at this, but we find that that variability on the order of a few kb does not actually significantly influence our estimate, but it is something that we can actually try to estimate the uncertainty of using empirical data. So that's a good idea to pursue.
In the interest of time, I'm just going to ask two more questions, but there are quite a few I'll ask you to answer offline, except online. So the first is from Eric Lai, who asks, hi, Athma, wonderful work. Is there any difference in the cleavage rate that might correlate with 3' UTR length? Or do you think other features of gene exon models, the length or the numbers, or do you think the cleavage efficiency is decided very locally?
So we're still trying to really go in depth by bringing in other data sets, such as RNA binding protein data sets. As I mentioned, looking at splicing upstream of the actual cleavage site, and also perhaps mRNA stability estimates to understand and address that question a little bit better. I do think that it most likely is going to be a combination of both a local decision of the rate at which you're cleaving at a particular polyadenylation site based on the local sequence context, but also a less local decision based on the other conditions of the gene, such as the expression level of the gene, or as we observed for mRNA splicing, that there might actually be a gene level influence on the rate at which regulation is happening. And for mRNA splicing, what we observed is that different introns in the same gene tend to be spliced at a more similar rate than introns that are randomly selected from different genes. And that might also be occurring for the rate at which particular polyadenylation sites are being cleaved as well. But I don't have an absolute answer for whether or not that's true yet or not.
Okay, we're going to stop there and let you answer the rest of the questions through the question and answer window.